Yeah, so you should be able to just click that. I don't think we have to do all the intro again just about what SARS is. We know that now. Let's see. So let's maybe jump in here. So coronavirus spike protein is the primary determinant of viral tropism, is responsible for receptor binding and membrane fusion. It is a large, approximately 180 kilodalton glycoprotein that is present on the viral surface as a prominent trimer, and it is composed of two domains, S1, S2. The S1 domain mediates receptor binding and is divided into two subdomains, with the N-terminal subdomain often binding sialic acid, and the C subdomain, also known as the C domain, binding a specific proteinaceous receptor, and I think that would fall into the neuropilin receptor. So we'd have to go back and check and say all this molecular stuff. Not my, it's not my area. I mean, I can pick bits out, but I'm not the expert. So anyway, the receptor for SARS-CoV has been identified as angiotensin converting enzyme, similar to what was been recently identified with SARS-CoV-2. HeLa cells transfected to express or not the ACE2 receptor from either human, Chinese horseshoe bat, mouse, civet, and pig were infected with SARS-CoV-2, and it was reported that the virus was able to use all receptors except mouse ACE2. SARS-CoV-2 was not found to use dipeptidyl peptidase 4, DPP4, the receptor for MERS-CoV. Following receptor binding, the S2 domain mediates viral conserved fusion peptide. The fusion peptide is activated through proteolytic cleavage, a site found immediately upstream S2, which is common to all coronaviruses. In many but not all coronaviruses, additional proteolytic priming occurs at a second site located at the interface between S1 and S2 domains. The use of protease in priming and activation to the S1, these domains, the S1, S2, that's the furin cleavage site. The use of proteases in priming and activation, combined with receptor binding and ionic interactions, hydrogen and calcium, together control viral stability and transmission and modulate the conformational changes in the S protein that dictate viral entry processes into host cells, specifically SARS-CoV and MERS-CoV. Both infect type 2 pneumocytes in vivo. However, they individually infect ciliated bronchial epithelial cells and non-ciliated epithelial bronchial cells. So this is basically, they're talking about different cultures that they're growing on, and you say that we can jump a bit ahead. And it's all talking about comparing the different strains, and we know there are different strains. So in this study, we performed phylogenetic, bioinformatic, and homology structural modeling analysis of SARS-CoV-2 in comparison with closely related viruses. We identify a distinct 4-residue insert featuring 2 arginine residues that map to the S1 and S2 priming loop of SARS-CoV-2, which is missing from other SARS-CoV related viruses, but present in MERS-CoV and in many other coronaviruses. We discussed the importance of this extended basic loop for S protein mediated membrane fusion and its implication for virus transmission. To obtain an initial assessment and shared and/or specific features of SARS-CoV-2 spike S envelope glycoproteins, protein sequence alignment was performed to compare the sequence of the Wuhan Hu1 strain, the novel coronavirus, with that of closely related human SARS-CoV-S strain. 4-2 sequence of journals should not be allowed to use supplementary figures. Sorry, it's really annoying. The overall percent of protein sequence identity found by the alignment was 76%, figure 1. Breakdown of the functional domains of the S protein based on the SARS-CoV S sequence reveals that the S1 receptor binding domain was less conserved, 64% identity, than the S2 fusion domain, 90% identity. Within S1, the NTD was found to be less conserved, 51% identity, compared to the receptor binding domain, RBD, 74% identity. So I guess what they're trying to say is that SARS-CoV-S SARS-CoV-2. First, I missed a one. So SARS-CoV spike. So it's talking about the original SARS, I guess. Yeah, okay. So what was that? S strain. Anyway, it's not clear, but it's not that important for this. A breakdown of the functional domains, we did that. And the relatively high degree of sequence identities for the RBD is consistent with the view that SARS-CoV-2, like SARS-CoV, may use ACE2 as its host cell receptor. Yeah, it's kind of old news. And interestingly, when the more defined receptor binding motif was analyzed, i.e. the region of SARS-CoV-S containing residues that were shown to directly contact the ACE2 receptor, the identity between the two sequences drops to 50%. So basically saying that there's a bigger difference between SARS-CoV-2 and SARS, and I'm guessing the SARS-CoV-2 has better homology to the human receptor. Yeah, we here in Cambodia, 1 million cats are eaten. Yeah. This is present a greater risk. Yeah, my advice right now would be like try to eat chickens and beef and pigs, maybe. Not not carnivorous animals are things that you should probably avoid. Da, 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 da. Oh, we're getting up to nearly at the magic 300. Now my Facebook is open for other friends. I don't lots of bonding going on in the chat. I like to see it. So I got 50 people watching. Damn, you guys are gluttons for punishment. So as expected, within the well-conserved S2 domain, subdomain identities were high for the fusion peptide region, high for the heptad repeat 1 region HR1, HR2, and high for both the transmembrane and the C-terminal N domain. Let's see. Early phylogenetic studies on SARS-CoV-2 genomic sequences revealed that it clustered closely with sequences originating from SARS, like sequences from bats with lineage B of beta coronavirus genus. Yeah, we know all this. Lineage A groups, blah, blah, blah. Hepatitis virus, and human coronavirus, HCOV-HQ1, OC43. The other highly pathogenic coronavirus, MERS-CoV, found within lineage C. Okay? Along with related camel-derived MERS-CoV, lineage C also groups viruses from bats and other mammals such as hedgehogs. I want to it's the paper that was in PNAS this week seems to suggest that species crossing is not as prevalent as they would what they thought. And so anyway, what are we looking for? So here's alpha coronaviruses, here's beta coronaviruses, and Here's RATG13. That's the one from Wuhan Institute. And where is SARS-CoV-2? So here's the SARS-CoV-2. And here we've got SARS-CoV-2 USA, Taiwan, Shenzhen, Canterbury, Foshan, Guangdong, Guangzhou, Zhejiang, France, Wuhan, different countries. So basically saying that it's closest related. We know that it's related to the RATG13, and we don't know if it's a modified RATG13. Who knows? Comparative analysis of SARS-CoV-2 as protein sequence. Protein sequence identifies identities between SARS-CoV-2S with SARS-CoV-S. The S protein sequences were aligned using MAFT, and the sequence identities obtained for full length and domains and subdomains are shown on the S protein diagram. Amino acid numbering, delineations of S protein sequence, 15 isolates of SARS-CoV-2 was aligned using MAFT with representatives of all 4 beta coronavirus lineages. A maximum likelihood tree was generated based on alignment. The tree was routed using the alpha coronavirus HCOV229E sequence. Of two. Highly pathogenic beta coronaviruses SARS-CoV, MERS-CoV are highlighted bold font along with bat SARS-like coronavirus closely related to SARS-CoV-2. That's that group there, and session numbers of sequences used in analysis found. Well, that doesn't matter. So CoV2S sequence clustered very closely with bat SARS-like sequences, with the closest matching sequence corresponding to a bat coronavirus. Bat CoV strain named bat SL RATG13 is the one from the Wuhan Institute of Virology. Other closely related sequences found were from bat SL CoV ZC45 and bat SL CoV ZX C21. The subclade that groups SARS-CoV-2, bat SL RATG13, bat SL CoV 45, and the bat SL CoV Z is distinct from one grouping, human and civet SARS-CoV, along with other related SARS-like virus such as bat SL LI-A3. Well, bat coronaviruses are the established reservoir for SARS-CoV-2. Presumed intermediate host remains to be determined. It's the Wuhan Institute of Virology. Based on the high degree of sequence identity in the receptor binding domain of sequences found. No, they're going down the pangolin one. I've lost interest in this paper now. It was proposed that these mammals could be intermediate hosts. Subsequent whole genome analysis revealed 85 to 92% identity SARS-CoV-2, which is less than what is observed for bat SL RATG13 96.3. To explore the relationship of pangolin CoV to SARS-CoV-2, we performed a phylogenetic analysis of the spike gene, pangolin genes, isolated from animals smuggled into China in 2017, bound for the super pots, no doubt. So we're trying to understand here S full-length identity between SARS-CoV-2 and pangolin, and they're trying to look at is the blue is 1, 2, 8, 5. Is that the whole you want to look at? And differences domains. The receptor binding domain is different. So where on the tree does it fit? Pangolin, bat CoV-2, and I don't know why. Why should this sit closer to this clade than this one, rather than RATG13? Odd. see, so it's this that I really wanted to get to, and right. So alignment of receptor binding domain and cleavage sites of SARS-CoV-2 and other bat COVs. An S protein sequence alignment focusing on the RBD region of SARS-CoV-2, SARS-CoV, and bat SARS-related viruses revealed that the N-terminal half of the RBD is relatively well conserved, whereas the C-terminal half, which contains the receptor binding motif, exhibits more variation. Notably, BAT-SL-CoV ZC45, BAT-SL CV ZX21 both have 2 deletions of 5 and 14 residues within the RBD. Composition of residues found at the 2 known coronavirus S cleavage sites was performed. Alignment data, region around the arginine, SARS-CoV-S, the S1-S2 cleavage site aligned well with SARS-CoV-2, the BAT SARS-related sequences. And this is a bit that's interesting. Notably, an arginine at the position corresponding to SARS-CoV R667 served for the other 5 sequences analyzed. The alignment shows that SARS-CoV-2 contains a 4 amino acid insertion, the PRRA insertion, that is not found in any other sequence analyzed, including the closely related BAT-SLRATG13. And remember, it's this little insert that gives it the capacity to bind to and attack the cholinergic receptors. So in this S protein, we've got 2 what are out of place gain of function regions. So the furin cleavage site between the S1 and the S2 domain. And then we can look down here, and here it is. So the S1, S2 is what we're interested in, really. I'm not, I don't think that much. Well, yeah, I guess saying these are different for the receptor binding domains, but so we know that it was how would you say the phylogenetic studies and binding studies have shown that it's tuned for primates, right? And so here's the S1 S2 cleavage site. So we've got SARS-CoV, which is the first one, SARS-CoV-2, the BAT RATG13, and all the others, and it's only the SARS-CoV-2 that has the PRRA insert. That's the binding region for the cholinergic neurons. That's, I think, what's causing the wide disparity in symptoms. And I'll put dollars to donuts is the reason that people are losing their hair, because nicotinic receptors control the little muscles that help your hair stand on end. And it's called piloerection. And I'm guessing that, and so importantly this morning, we looked at a study where even though someone tested negative in both swabs and antibodies, they found traces in the skin. So biopsies of skin showed that they were infected, and it would make sense if this virus can, like I say, it's got the ability to passage orthograde and retrograde down nerve fibers, and it targets the small muscles at the base of the hair. And maybe I should probably just Google and cholinergic. Just check I'm not completely bullshitting you. So, Recon. Yeah, my old institute. I've got things to say about them. It's not ready yet. see. Well, anyway, there's small muscles at the base of the hair, and it stands the hair up, and if it's up, it's trying to keep you warm, and and all hair, I think, has it, even the hair on your head, and it's a small muscle. And let's look for choline. Okay, so transplanted hair follicles were responsive to acetylcholine, form proper connections with the surrounding host tissues. There you go. So there we've got cholinergic mechanism at the hair follicle. Okay, and if it gets attacked by a virus, it's likely damaged. That's why your hair is going to fall out. And I would imagine it's probably going to be a permanent state. So Philiton says Dr. Paul Cottrell has done a video of HIV GP41 found in SARS-CoV-2 spike protein. Yeah, I watched it. It wasn't particularly compelling in any way, and the it's not. It wasn't even in the spike protein. I think it was in uh, one of the N surface proteins. This be the same aspect that makes one's tartar shrink. Would that be the same manifestation on women? Well, it's the autonomic system that does this, and you've got to be well. You know, it's basic biology to understand the mechanisms of what that system does. Right. So one is called the parasympathetic branch. It uses choline sort of externally or in the peripheral nervous system, and then it projects, and then its next axon is usually noradrenaline, and it becomes a excitatory branch of the autonomic nervous system. The vagus nervous system causes the so parasympathetic is fast or speeding up, and vagal is slowing stuff down. Hair falls out from COVID. Yes, and I would say that this is looking at why that happens. Okay, that what now we've localized that there's a acetylcholine like response to the virus. Now we can begin to tease apart the symptoms and say which components fit what functional aspects of the disease itself. And I think the ACE2 binding site, obviously, particularly the lungs and the the smooth muscle, but we know now that this cholinergic assault is going on, and acetylcholine is also involved in modulating the immune system and modulating the platelet response, right? So that in the absence of acetylcholine, because the PPRA insert acts as an antagonist at the cholinergic receptor, it causes an imbalance in the immune system, and that potentially leads to the cytokine storm, and that's why smoking those freedom torches seems to have been somewhat beneficial to the uh, patients at large. Give bio, maybe I'll take me on 6. Where do dumbass people keep your hair? You know, um, I'm all for you suffering the ignominy that I had to. Um, that's not fair because the hair loss for women, I think, is much more traumatic, and even though as a man, problem. Anyway, let's get back to our paper that I want to listen. So, uh, so off. All right. So yeah, I basically just wanted to get to this, and it's another confirmation of this PRRA insert. They don't really talk about it, but they're more interested in the furan cleavage site. But let's see if we can see anything approximating a discussion here. These are beta coronavirus Wuhan. They have PRRA. Beta cov pangolin doesn't have it. That SL. SARS cov doesn't have it. So someone put it in there, right? And that the furan cleavage site, if there is elements of, well, this sequence is synonymous with HIV GP120, and again, don't know. We don't know what tissues these were passaged through. We don't know what the specific aim was. I don't. It's not vaccine research, right? Otherwise, that you'd probably be doing like one thing at a time, and it would probably be almost attenuating it rather than um, doing what we've seen here, which is a very particular sequence that, like I say, it's once you're mimicking rabies, okay, time to sort of pull your head out from the bucket and take a look what's going on. You were saying about HIV GP41 code wasn't that compelling. Why would that be? Because I don't think it was part of the S protein. It's part of. If you look at, I mean, it's interesting that it's there, but it's a small repeat. But it's not part of the. It's more part of the membrane structure around the glycoprotein. Let me quick. Is it possible to add this sequence within a lab? Um, Presumably. I mean, it can be added where it was done. I don't know. This is a good link. I highly recommend this one. So this shows you all the proteins for SARS-CoV-2, and the SARS-CoV-2 genome, Alpha-1AB protein, structural proteins, and he was looking, I think, at this protein, okay, and this is the spike protein here. So if GP142 is in this part of this sequence, yeah, I mean, it sort of indicates that there's been manipulation going on. I don't need convincing of that fact anymore, right? The PRRA insertion and its obvious increase in superantigenicity and the furin cleavage occurring at the same time does. I mean, it's something that virologists can argue over, but from my perspective, as soon as it's targeting the cholinergic receptors, it becomes a systemic neuroscience and neurology issue, and the virologists probably should just shut up for a bit and let us get on with the job of looking at the symptom profiles and ways to interact both with cholinergic systems, blocking of the ACE2 binding, and yeah, I don't know. You need to come up with some sort of small molecule type therapy. So anyway, this document will show you all the proteins. NSP1, I think this is the one what was going on about. Protein shows cell's production of its own protein. This sabotage forces the cell make raw virus proteins, prevents it from assembling antiviral proteins that could stop the virus. Cellular saboteur. Mystery protein NSP2. Scientists aren't sure what NSP2 does. The other proteins it attaches to may offer some clues. Two of them help move molecule-filled bubbles called endosomes around the cell. Not the endosomes, no! Untagging and cutting NSP3. NSP3 is a large protein that has two important jobs. One is cutting loose other viral proteins so they can do their own task. Also alters many of the infected cell's proteins. Normally a healthy cell tags old proteins for destruction, but the coronavirus can remove those tags, changing the balance of proteins and possibly reducing the cell's ability to fight the virus. Remember guys, that this virus has the capacity to change the cytoskeleton of the of a cell such that it will send out tentacles and punch holes into a neighboring cell. Bubble maker, combining with other proteins and as before, helps build fluid-filled bubbles within infected cells. Inside these bubbles, parts for new copies are constructed. Protein scissors, NSP5. The protein carries most of the cuts. Blah blah blah. Bubble factory. Yeah. Copy assistance. Nice. These two proteins help NSP12 make new copies of the RNA genome and ultimately end up inside new viruses. The heart of the cell, NSP9, this protein infiltrates tiny channels in the infected cell's nucleus, which holds our own genome. It may be able to influence the movement of molecules in and out of the nucleus, but for what purpose, no one knows. Well, I would imagine you want to stop the host cell being able to send out RNA that would be used to... Well, we know that there was the paper that showed that SARS-CoV-2 blocked the major histocompatibility complex. So that's a molecule that if it finds something foreign in the cell, it grabs it, puts it out to the cell and says to the immune system, please, there's something here, come check me out. And then it decides whether you're going to get eaten or not. Endosomes are in, exosomes are out. Yes, very. Yeah. Know that HIV code is found in Ebola glycoprotein chain B. It won't surprise me. There are similar sequences, right? So you can look at a chimpanzee and you find similar genes in the chimpanzee. So I'm not sure how much it means if there's... It depends on the functionality, right? Now the PRRA sequence has a very direct effect on the pathogenicity of that agent, right? So it's not just going for the lungs and the ACE2. Once you're targeting cholinergic receptors, that's it. You're in the brain everywhere. You're getting into the muscles, the skin. We're seeing that the skin is a reservoir now, and you've got to. Well, the other thing is not to read much into. But anyway, it makes genetic camouflage. Interesting. Copy machine. All these sequences, and these sequences, funnily enough, are unique to virus, and that's what gives it its classification. And it's not part of the human genome, right? And that's one thing, right? If I get to meet one of these people that thinks that viruses aren't real, then they have to presume that the a case report for rapid onset brief psychotic disorder. She turned into a COVID zombie. This is not... shouldn't be new to us, right? We should be expecting this. And what I would see renovation. Everyone, please make sure that the papers go into folders. I do try to an RNA virus compelled to put in a viral cell has DNA in it. I don't know. Into a reservoir, would having your face tough be risky? I don't know, dude. Just having your face pulled off would be risky. I don't know, BP, to tell the truth. I dread to think what will happen in 5 years' time. Well, look, I would say this with what appears to be a very obvious move with respect to next generation warfare. I think that's what we're dealing with here. The biologicals are in play right now, and we have to see what that really means and how many are they going to take out before there's real declarations of war. I'm very much hoping that Brigadier Spalding will really be able to give us some insight into that, because it's been known for decades that the Chinese were just lifting tech from the United States, right? Even before the Clintons, there was huge issues, and they are well, they the Asian countries are based, right? And if you're part of a different tribe or country, they let you know. And Japanese don't like Chinese, Chinese don't like Japanese. Same with South Korea and all their neighbors. They all hate each other. um, which is surprising, because you seem to like you come here and they do seem sort of somewhat organized, but I don't know, man. They've got very long memories about what to me with ACE2 receptor after the virus infects one cell, if it can have cell that, yeah, once it's in, the tentacles allow it to evade the immune system, and yeah, you don't need you don't need other receptors if it can sort of physically bind. I don't know. I mean, we're making assumptions. Maybe it does. So I think I'm gonna leave that there. Well, let's show their figures off, but not really sure what you're going to get from that SARS-CoV structure. SARS-CoV-2 S model. Well, so there you're looking down on top side. And we know that the PRRA juts out from this sort of angle here, so it's orthogonal to the top receptor binding domain. That is. I want to get a conclusion from this that's somewhat satisfactory. I don't want to hear about. I want to hear about this PRRARS and anyway, okay. So the current COVID-19 pandemic, blah 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 blah. Identity of the S protein at the bat RATG13 to bat coronavirus was shown to have obviously the most similarities, suggesting the virus is the closest relative to SARS-CoV-2. While the origin of the novel coronavirus appears to be in bat reservoirs, there is still no definitive evidence of the possible intermediate host to transmit the virus to humans. No, it's just humans passaging cells through cell lines and having the virus do all the hard work for you. Recent reports have suggested that Malayan pangolins. No, we're not listening about pangolins. Sorry, that's busted. In this study, we show the presence of a distinct insert that maps to the S1 S2 priming loop. We know that, and is not shared with SARS-CoV or any other SARS viruses and be it coronavirus lineage. Yeah, so that's a bit just that thing alone, because it's so unusual with respect to its immediate uh, phylogenetic tree, should be enough to make people say, whoa, what's going on? Now we've got that plus the PRRA insert, which binds the cholinergic system. That means you've got two gain of function elements very, very close to each other, and each can attack critical components of the body. Right, that SARS-CoV receptor binding motif down conformation packs more closely the NTD of the S. Interestingly, we observed in our models that the receptor binding domain is predicted to pack similarly to SARS-CoV. The RBM is also predicted to organize as a flexible loop, similar structure between the lower amino acid identity of these two proteins. A more recent report has been shown that ACE2 binding mode of both SARS-CoV, SARS-CoV-2 RBDs is nearly identical, which supports the claim that the flexibility in the RBM is key to compensate the amino acid differences between the two CoV proteins. Agrees with our predicted models. One of the common difficulties of the CoV-S protein cryo-EM studies is the difficulty to solve proteolytically sensitive regions in the protein. Since the S1, S2 region is proteolytically sensitive, it is common to introduce mutations in this site to prevent proteolytic priming, to allow efficient heterologous expression and purification. This has resulted in the difficulty to solve the S1, S2 region in most of the available CoV-S protein structures, with a few exceptions. Long. Well, for me that was interesting because it's got the PRA sequence in it, and there's nothing else really new in that. So what can we go through real quick? So let's have a look at this one. Case report, so it should be free. 36-year-old previously healthy woman, no personal family history of mental illness, presented with new onset psychosis after a diagnosis of symptomatic COVID-19. Her psychotic symptoms initially improved with antipsychotics and benzodiazepines, and further improved with resolution of COVID-19 symptoms. First case of COVID-19 associated psychosis in a patient with no personal or family history of severe mood or psychotic disorder, presenting with symptomatic COVID-19. I remember I was talking to you about the other day about the robustness of brain networks, and you would be surprised how often you can stick probes into the brain and you don't notice anything. So there's this reserve capacity, and what I would argue with this potential that we're seeing these psychotics, right? So generally, if most people are around the median around here, and then this woman generally is up here, and it's not a problem. She's not in the danger zone. But if something happens, a bit of stress, boom, over she goes. And I would say the brain took a bit of an insult. Probably the well, who knows? I mean, we know that there are multiple factors at play, including the what's it called? It's demyelinating syndrome. I forgot the first bit, but and also the retrograde travel up the nose and into the limbic system. Don't forget that one. So, did she have encephalopathy? The African American woman. Yeah, and it might be the case that you're going to see prescriptions going up for these drugs, the atypical antipsychotics, as people recover from this illness, right? And like I say, I have noted a distinct degradation in my left side because of head injury, and after the COVID bout, I definitely got more motor abnormalities. notice that, and my meds definitely aggravate it. So you know, that's getting old. You got to get on with it. So is there any virologist out there you trust? Maybe we can get a virologist on board in Discord and here to help us understand. I'm happy to speak to any of them. The issue I found is, people want to speak in their own main, and they'll start arguing. Like with virologists, they'll say, "Yeah, but if you look at this amino acid and the odds of it flipping to become another one," and they it always just sounds a bit sketchy to me. But yeah, it would be nice to have someone who knew the ins and outs of these protein structures, right? And what could be their potential effect to how they would change uh, under environmental pressures. So anyway, our knowledge, this case prevents first description. Yeah, lots of people had. When was this? Finding dates on. But I think so. I don't have anything massively interesting else to look at. We've gone through. It was a bit more serious stream than I was expecting. Actually, we tried to cover three papers, and these are things that we should be more and more familiar with as we have um, combed through the literature, tried to find coherent and parsimonious explanations for why we're seeing the response that we do. And so, what I might do is let's numbers. I looked at Worldometer this morning, right, and it seemed to me that we'd hit a plateau of 20 million, 21. Right, it's flattening out, which is good. Right. Um, Now we need to. Well, I don't got other countries to take into account. Africa, Middle East. I would be. I would be skeptical, but that is what's the date there? August 14th. Wow, so nearly 3 weeks, 4 weeks. Been holding steady. That's interesting. Well, it did it down here as well, so I don't know if we're going to see more surges, but um, you also have to remember that at this time, it started going back up as they opened up, right, and they've shut back down again. And yeah, what do you do in this circumstance? I don't know. I don't think the stopping of society for everyone is a good thing. A lot of people can't handle it, and a lot of people are snapping. And the there are people running around out there with that virus in their head that are having psychotic episodes like that nurse, but she had healthcare. Imagine the nuts that are under bridges and stuff. That's just part of their repertoire, right? And they're just somewhat somewhat more florid on that day than they were the previous week. So I guess if someone's got something they would like me to look at, that do 2 hours streaming. So I might call it a night It's 1:33 a.m., an early night for me, and I will try and think about something to do. I don't know if I'll stream tomorrow. It's Sunday, and kids go back to school Monday, so good. Got 66 people watching. That's great. But YouTube chat is broken. Thank you. 18. So nice info, Kev. Many thanks. Welcome. Vitamin C, D, zinc today. Good. 5G rolls out. It will increase again. Exosome storm incoming. Yeah, I don't think so. If they're having this headache, people get cranky. Yes, I would imagine. It's only gone up where it's not flattened or only increased. Only gone up where AM not flattened or only increased. Ecuador and Bolivia have a lot of mass graves. Yeah, so do Guatemala. Endemic now, indeed. Yeah, plus more testing too. Remember that the heavy metal band in the... No, actually, you know what? I saw graphs that testing has dropped off significantly. Where did I see that? I want to say it was on the Reddit thread. That's not Reddit. It is 1:30. On. Hot Russian doctor quits over questionable ethics approval that rushed through coronavirus vaccine. Yeah, and the same applies to West's vaccine programs right now. It's not being done scientifically, not for what they're trying to achieve, and I, I don't know how many more times I can say it because it just becomes like you're speaking to a brick wall. CDC director warns America is in for worst fall we've ever had. Only a month or so away, right? That's 6 weeks. Fauci believes normalcy will return by the end of 2021 with a coronavirus vaccine. This is another thing that I understand the people, right? The mask thing, I was just like, come on, man, don't be so stupid, right? When there's something going around where it's raining outside, put a hat on and a jacket. Same with the mask. But I'm on board with people that are upset about the idea that the government can mandate that they have an experimental vaccine jabbed into them, and there's no liability to the companies that made it. Right? That's an absurd state of affairs to be in. It basically just shows you that you're chattel to these people, and the sure as shit they need to find a method that isn't injected. Right? It needs to be like drops or something, or I don't know. They had like GMO tomatoes, right? Um, they need to do something that is more less invasive and dystopian. See. Daily coronavirus cases reach 5-month high. What's going on, South Korea? Try Miro to ideate on a virtual whiteboard. Share feedback. Why is that not working? Scientists. Event. clearly not doing enough. Drop in. Yeah. So I saw it. Cool. So maybe I can. Month, public health experts and federal officials have said that significantly expanding the number of coronavirus tests administered in the United States is essential to reining in the pandemic. Sorry. By some estimates, several million people might need to be tested each day, including many people who don't feel sick. But the country remains far short of that benchmark. For the first time, the number of known tests conducted each day has fallen. Reported daily tests trended downward for much of the last 2 weeks, essentially stalling the nation's testing response. Some 733,000 people have been tested each day in this month, on average down from nearly 750,000 in July. 7-day test average dropped to 709,000 on Monday. Picture of this to graph it. But point being, there's there is a drop in testing, and so obviously the number of cases drop. Now, I guess what happens then is that there becomes an increase in death rate versus total cases. So that would impact the case fatality rate, but I don't. The case fatality rate is a useless ratio right now. Do not use it. Closed cases. That's that done. And let me check. I have to just.
Now, each day we use data from local health officials and the state.
See if there's anything in my feeds. Police officer making an arrest never ever leans against the wall while describing charges. You stand upright on your toes, ready to deal with any possibility. I don't know what this Millie Weaver thing is. It's another American thing going on. Some things are hard to bring up motivation for. I think Arms would have been there by now. Legs will float its way there. Burgle, I don't know what you're talking about. Like one step.
And gushy, make that pull out game weak. Yeah, you dealing with some wet and gushy. Bring up, I said certified free. 7 days a week.
Man, if there's anything that signifies the collapse of a civilization, it's the emergence of the rap hip hop music scene. Nothing is more base than animalistic than oh dear, what's this? Date. Okay. CDC backtracks guidance on 3-month window of immunity. Okay.
From the Centers for Disease. A person who has recovered from COVID-19 will likely be safe from reinfection for 3 months, according to updated guidance from the Centers for Disease Control and Prevention. The information marks the first acknowledgement of a defined immunity period for people who have recovered from a COVID-19 infection. Prior research has shown antibodies from recovered patients will fade over the course of a few months, but federal scientists had not previously said what that means for immunity.
Well,
oftentimes Black businesses are totally ignored.
I think the issue is that it's not antibody dependent, right? It's the B cells. So the issue is not just survival. My son and I, no prior health problems. Both had heart trouble for weeks after COVID infections. Please knows it attacks the organs, heart, lungs, and more. Its symptoms were ghost white face, grey under eyes, at times red around eyes, tired aches, keep activity low, and watch heart rate. My boy's been like that. Is happy of yeah, so I think I'm done done with stream, guys. I don't have anything much more to add, so honest, no helps we don't close and getting inside there. Is that what the Danny DeVito character was doing? Liquid detergent. I don't know, dude. That's the biopsy one, and I forgot what I've done with that one, but